Okay, now it's recording. You had a question? Okay. Good. So the lecture will be recorded the next morning on even 30 minutes after the lecture. It'll be available through UJA. And if you have problems accessing to UJA, there is always another method, okay, through the Canvas, and I can give you that information. And the exam is ExamSoft with exam ID. And it's Canvas, the module home usually has that information, but it's yet incomplete. A little bit of a modality, the ExamSoft, will be completed as we go. Okay, Sylvie, you want to say
Yeah, absolutely.
anything?
I don't know if everyone in the class can hear me, other than those that are logged into Zoom. So I apologize for that. But it is recorded. So yeah, my name is Silvio. I'm a third year medical student, and I'll be helping out Dr. Friedenreich and Dr. Ivesa. I'll also be sending out an email just with kind of like the summarized info that I'll share today. So I'm just gonna share a couple quick tips. Biggest thing is not to fall behind on this lecture, on any of the lectures. Even though you only have one every week, uh, it's easy to just fall behind. And the best thing is to review right after the lecture or the next day. I have a lot of resources too, like I have an Anki deck I made. There's also my own notes I made before. Some of them still apply, most of them still apply, but for some of the newer lecturers, like Dr. Mojica's, you guys might have to change a couple of things. But again, I'm always able to answer questions on GroupMe or email, so feel free to email me. I'll respond as soon as I can. If not by the end of the day, the next day for sure. I also advise reaching out to the lecturers. They obviously know this way better than I do. But again, if there's anything that's like concerning or just like you really don't understand something, again, feel free to put it in the GroupMe. I'll get to it. Sometimes your classmates will get to it, and that helps a lot as well. There are no office hours for this, so it's really more so just you guys really asking questions if there's something that doesn't make sense. I unfortunately don't have access to the exam, so I don't know exactly what they'll ask, but I have an idea just from previous years of doing it and just kind of students talking as well. As mentioned, the grade is based only on two exams, so it's 50% each of them. I know that's kind of terrifying, but it ends up working out really. If you don't do well on the first one, you have the second shot, and depending on how the class does at the end, there is kind of a curve, we can call it. It's really just the grades are adjusted depending on the grade scale. Again, we don't know what that number will be, so it really does come to the very end. So that's why I always say just to stay on top of the lectures. They're long lectures. Number of slides changes between each lecture, and again, it's a lot, but it's everything that you're tested on will be on the lectures. So it's not something that you have never seen. That's why he's mentioning finding, YouTube videos or going to the library. That's only for you to really understand the concepts more, but don't focus on outside resources for testing because everything that you're tested on will come from the slides directly. So there's no tricks really, and that's something that some students kind of forget and go off of YouTube, like what they said. And like I said, it will come from the lecture slides, every single question really. There are no quizzes, but I do try to release quizzes that I make. Sometimes some of your classmates will make quizzes too, and it's more so just to test your knowledge. And again, if there's something that you don't understand in those quizzes, reach out to me and I'll help you out. We'll get through it. So in the Canvas itself, you'll see a TA module, and that's where I'll release all the resources that I have for you guys. And then once we get to the review itself, the review will be held on the same lecture day. It'll be a week before your exam. It usually takes about an hour and a half, max 2 hours. And we'll kind of just hit very high yield points for each lecture. And then I'll finish with about 20 or so questions from the review. And that's the review itself. And obviously when we get to that, we'll talk about it a little more and I'll release some resources for that, just so you guys are prepared. Again, everything will be recorded. If there's an off chance that I am able to hold like an office hour through Zoom, I'll let you guys know beforehand. Um, I don't know if I'll be able to record that or not, but anyone who can make it, great. If you can't make it, don't worry about it. It's not that you're going to miss anything, because again, I don't have any of the questions for the tests at all. Um, so far, does anyone have any questions though? Somebody
No questions here.
Somebody in the group mentioned if there you guys have access to practice exams. There are no full-length practice exams, but I will have like a giant folder with all the quiz questions that I've made up. Some people find that very helpful. Others maybe not. They just kind of rely on the lecture. So it really is up to everyone, kind of what works for them, what doesn't. But you will have a kind of practice exam quiz type thing that'll be on Canvas. It'll just be a document with questions, and at the end you'll have an answer key. Yes, all the questions on the exam will be multiple choice. There's no fill in the blank, and it's just 70 multiple choice questions.
All right. Anything else or
No, I think that's
ready to
pretty much,
start? Okay,
yeah. And then again, if anyone has any questions, just email me or send me a message to group me, and I'll send out a message or an email with all my information that you need.
very good. All right, thank you. Thank you so very much, Silvio.
Thank
And
you.
I'm telling you, go to the review sessions. These are very, very important. Okay. Couple of, I think that I covered mostly everything. I have my cheat sheet. The recordings will be available through UJA, and the information about the exam soft is will be provided as we go. The curve is usually, we try the curve to be generous. But of course you have to really study for the exams. I want to emphasize there's a lot of information here. You may feel I went to one of the lectures, I didn't really worry. I think that I can handle that. But after 7 lectures with complete different topics, different lecturers as well, you may end up exhausted. So I strongly recommend you to study one by one. For example, after this lecture tonight, either tonight or tomorrow, you just take a look at it. If there's something that you didn't quite understand, write a list and clear those questions as soon as possible with the lecturers, in this case with me or with Silvio, okay? So that's the dynamics that we recommend to you. All right, we are going to start right now. If there's anything else to be done, no. Okay, so again, my name is Diego Friedenreich. I'm the director of the course. The co-director is Dr. Andrea Saifesa, and the TA is Silvio Giraldo. These are our email addresses. Okay. So this is the title of the lecture. We are going to give an overview of the organelles. This is an animal cell. You may have studied this many times. This is an animal cell, and we have the nucleus here. With the nucleolus, we have the Golgi apparatus, we have the ER, the endoplasmic reticulum. We're going to study them, and give you some overview about them. But this is the essentials of tonight. We have the mitochondria, we have the ribosomes here, we have a peroxisome, we have a plasma membrane, we have the nuclear envelope, the nucleoplasm, vesicles, and then the railings of the different organelles, which are the microtubules, that this starfish kind of structure. We have the actin filaments, and we have the intermediate filaments. And the animal cell doesn't end at the plasma membrane. There is extracellular matrix. There is secreted to the outside of the cell. So this is essentially like our baseline, our main system to study. All right, the two main characteristics or features of eukaryotic cells, the ones that we are going to study, is exchange of information between inside and outside. Nothing stays inside forever. There's a lot of traffic. There's a lot of secretion. There's a lot of import. There's a lot of transfer of information between the cell and the outside. So that's one. The other one, subcellular compartmentalization. Sometimes in a cell, if you look under the microscope, you'll see some organelles located in different, for example, all of them at the basal pole. And that will give you a hint of what is the function of that cell. So structure follows the function. If you look under the microscope, a cell type, you'll make sure that this cell is gonna secrete some granules, for example, or it's gonna provide the contractile activity. All this can sort of be predicted by just looking at the structure. So we have the import and the export of a cell. There's a lot of import, there's a lot of export. Export or import as endocytosis. You can see all the particles here being imported, being phagocytosed, and at the phagosome here, essentially. And then export is the opposite mechanism, okay? When these granules or proteins or glycoproteins are to be secreted, they follow the opposite mechanism. That's a secretory pathway versus the degradative pathway. Okay, what's the origin of an eukaryotic cell? It's been claimed that evolutionary, the eukaryotic cell developed from a bacterium. The bacterium was small and had a very simple structure. Ribosomes at the plasma membrane, DNA at the plasma membrane, small. Turns out that over the years, there were changes, evolutionary changes. For example, the invagination of the plasma membrane here and the formation of the mesosome here. And that progressed a little bit more and formed what you have here, a structure that is a consequence of all these evolutionary processes. The nucleus with the DNA inside, which is protected by the whole nucleus, and also you have the ribosomes, but the ribosomes only at the outer membrane, plasma membrane of the nuclear envelope, and we'll see more details as we go. Okay, so there is like an kind of an asymmetric situation here. The ribosomes attached to the nuclear envelope, but only to the outside of the nuclear envelope. Okay, not the inner nuclear envelope, just the outer nuclear envelope. And then evolutionary, we have also another process which is very distinguishable. So this you can see, this cell is growing in size. It needs more energy. So at some point, during the prehistoric period, the bacterium was phagocytosed by this intermediate cell, right? And the bacteria has contains a lot of energy and produces a lot of energy. It turns out that this cell found appropriate or advantageous to have that bacteria within it. Okay, so the bacteria eventually decided to stay in this cell, which now has been transformed into an eukaryotic cell, right? So the eukaryotic cell has a nucleus, very distinctive, has DNA or the chromosomes within the nucleus, and it has the mitochondrion. Okay, the mitochondrion is the energy that keeps the cell going all the time. And it has been provided many thousands, if not millions of years ago from a bacterium that had been phagocytosed. Okay, a good reason for compartments is that an eukaryotic cell is a lot bigger than in volume than a bacterium. Okay, so it needs more organization, more structure, and that's why the membranes are being added to these organelles or to these elements here. All right, so to summarize this first part, compartmentalization is about more membrane surface to allow for more critical membrane activities and more organization, okay? If you have compartments, you have more organization and more critical membrane activities, meaning that plasma membrane is not just a separator between two compartments. It also has a function, and there are lots of activities, functional activities there. Okay, if you have more membrane, you have more activities. So that's one reason for compartmentalization. And then easy organization. It favors or it makes it easier for the protein to be produced and to be secreted to the outside. Okay, there is a mechanism to do that via Golgi, and vesicles. And there is an evolutionary energetic activity. It's sort of this intermediate cell ate up the bacterium and found out that, determined that the bacterium could stay there as a mitochondria, okay? But that over the hundreds and millions of years. All right, so this is just for you to get an idea of how compartmentalized is a cell. This is an electron micrograph, and it is showing you the complexity of this formation of these structures, where the information, the DNA, is at the center of this scheme within the nucleus, and then from there is an execution of imparted orders and the production, which is the enzymes and the proteins, the glycoproteins. All of those are the product of this translation of the transfers and the messengers, etc., etc. So it is very, very well organized and structure a cell. And the organelles that you can appreciate here, they're not just hanging in there, they are attached to structures. For example, microtubules, and a vacuole or a lysosome will travel through the railing of the microtubes to the right place. So nothing is random in a cell. There's a lot of pre-organization, okay, and a structure. This is just to show you that you can have, for example, spherical lysosomes or vacuoles, or you could have structures that contain a lot of plasma membrane, like this, okay? There's much more surface in these structures. In the ER, for example, 60% area of membrane is provided by the endoplasmic reticulum, okay? That's a lot of surface. The plasma membrane, it's 2%. Just compare 60%. Everything is sort of covered with different plasma, different surface, okay, at the ER. We have, for example, the nucleus providing 10% of volume. We have surface here and volume at the surface at the right, volume at the left. And it doesn't really always correspond volume and surface. For example, the ER or the endoplasmic reticulum has 20% of volume, just a lot, but 60% of surface. Okay? Same for the mitochondrion, it's 10% of volume and 20% of surface. And in some cases, it's 6% volume because it's spherical, okay? And 2% of membrane. So it varies a lot and it also depends on where the cell is, coming from, okay? All right. We are going to go over the nucleus. Remember that this lecture is an introductory lecture. We don't go into details. Then lecture 2, lecture 3, lecture 4, they are going to give you details of different parts of this lecture. So we are going to tackle the nucleus now. This is the structure of a nucleus with the nucleolus, which interestingly, it's been distinguished from the rest of the structure because it doesn't have any plasma membrane. It's not separated from the nucleus by a plasma membrane. It's what it's called a membrane-less structure or a membrane-less organelle, okay? That's the main feature of one of the main features of a nucleolus. But you can, when you look at the electron microscope, you can distinguish the nucleolus from the rest of the nucleus. So it's a membrane-less organelle. But most of the time, the organelles are separated by membranes. For example, here we have a nucleus. We have two plasma membranes conforming the nuclear envelope. Okay, the nuclear envelope is composed of two membranes: the outer membrane and the inner membrane. And there is heterochromatin here, which is attached to the inner membrane, and it is usually composed of DNA plus histones. It's heterochromatin, okay? And usually it's very compacted and conforms what you can see here as shadow here, and at the border of the nucleus. It binds to the lamin, which is also part of the inner nuclear envelope. So we have a nuclear envelope, the inner nuclear envelope. We have lamin, which is a structure that is attached to the inner nuclear envelope. And then we have the DNA, the heterochromatin, that is attached to the lamin and to the nuclear envelope. All this conforms the heterochromatin. And usually it's inactive DNA, DNA that is not transcribed. The DNA that is transcribed and forms and it's translated into proteins is this one, is the more laxly organized DNA without or with much less histones, so it can be in an open structure. It can they can be accessed, and there can be more activity with this DNA, the euchromatin, this one, okay? The euchromatin. Different cell types have a different proportion of euchromatin versus heterochromatins. For example, the stem cells have a lot of euchromatin, meaning that there's a lot of activity, a lot of transcription, okay? And there are other cells that do not replicate, they don't undergo mitosis anymore, and they have their DNA is silent, and it's composed mostly of heterochromatin, okay? So this is essentially what we have in the nucleus. We have nuclear pores, which are giant structures that will allow in and out different carriers. For example, DNA or RNA going, not DNA, RNA going out in a form of ribosomes, and proteins that have been synthesized and come in through the nuclear pores. So it's a very dynamic process, very dynamic. With the proteins going in, with the RNA or the ribosomes, part of the ribosomes coming out. And yes, finally they assemble into one ribosome, but outside of the outside of the nucleus, at the cytoplasm. Okay, so we have the nuclear pores. So I'll give you more details about the nuclear pores, nuclear envelope. Nucleoplasm, it's like the cytoplasm that we have outside of the nucleus and inside of the plasma membrane, but the nucleoplasm, okay? It's a structure that is inside the nucleus and where all these structures hang in. The nucleolus, as I mentioned before, this here, it's a membraneless organelle that facilitates splicing and maturation of ribosome RNA, okay? Splicing of every RNA or messenger RNA and the ribosome RNA maturation. That's the nucleolus. Sometimes there is one, sometimes there are two, there are three, depends on the cell type. Nuclear pores. Nuclear pores are it is observable through the nuclear envelope. This is an electron micrograph of the nucleus where you can see the nuclear pores here. They are giant structures composed of 50 to 100 different proteins that assemble one to another. And it operates like an old photographic camera with a diaphragm. The nuclear pore can sort of, depending on what is going to pass through, it will open and will allow some big complexes to come in or come out, okay? If the complexes are small, then the nuclear pore will be sort of on a close or a very, very tight configuration. This is how the nucleolus is being observed at the electron micrograph. And the nuclear envelope, a very, very important structure, is being observable here. This is the this is a euchromatin, and this is the heterochromatin. It's more refringent, okay, because of the histones and all the complexes that it's been attached to. All right, the complex, the nuclear pore complex, is asymmetric. This is the outer nuclear envelope, and this is the inner nuclear envelope. It is asymmetric, meaning that the outside part is different from the inside part. And it's organized in a way that it will allow one protein or one complex to come in. This is the description with the thick filament, with the anchor proteins here, the diaphragm that it's located here, something that it's called a basket, and it's part of a structure. It's an intriguing structure here. But for example, the proteins that are imported go from the cytoplasmic part to the nuclear part. And this is what I told you about the diaphragm, that it opens and closes depending on the size of the complex or the protein. And in some cases, if the size of the imported protein is very small, it doesn't require energy because it's mostly diffusion. They just go in by diffusion. But if that's a complex or it's a large protein, it needs some energy. For example, these blue and green complexes, they need energy to be imported. This is again an electron micrograph with the chromatin, the heterochromatin here, with the nuclear envelope. You can appreciate here the nuclear envelope, and this is very important. You have the outer envelope and the inner envelope. The plasma membrane that surrounds the cell, it's just one membrane. It's a bilipid member, but it's one membrane, okay? The nuclear envelope is made out of two membranes. That is an important distinction. It's the inner and the outer membrane. That's the nuclear envelope. And then the annulus here for the pore, the diaphragm, etc., etc. So this is how a protein is imported. Where is the driving force for the import? The driving force is observed or is localized to the protein. The protein has all the required information to be imported. For example, this one it's going to be imported. It has an NLS, a nuclear yeah, thank you. Nuclear localization signal, okay? That is recognized by the importing complex, alpha and beta. They bind to the nuclear, to the protein that has a nuclear localization signal, and they are imported, okay? Once they are imported, and you could see this, that it requires energy in the form of GTP and ATP, okay? This is essential for large proteins like this. And once it gets imported, it separates from the chaperone, the alpha and beta importin, and one of the two, the beta importin in particular, goes back to the to the cytoplasm, and then it operates over and over again. This is a very dynamic process with lots of histones that are coming in and lots of ribosomes are coming out. Okay, summary of the nucleus. There is a lot of protein import in the nucleus through the nuclear pores, and this is called gated transport. There are, for example, there are different types of transport. There is the co-translational transport, meaning that when a protein is being translated or created, at the same time it goes somewhere else, co-translational transport. There is also a post-translational transport, meaning that the protein is being made first, and then as a posterior activity, it gets imported or goes somewhere else. Or gated transport. The proteins that go through the nuclear envelope or through the nuclear pores are called gated transport, okay? That's an important distinction as well. For example, co-translational import at the ER, the protein is being made by the ribosomes that are attached to the ER. And at the same time, it penetrates the plasma membrane, it goes into the ER. So that's co-translational. For example, post-translational at the mitochondria. There is a complex TIM-TOM that facilitates that post-translational import into the mitochondria. And then we have vesicle-mediated transport when the structure that facilitates the import is loaded into a vesicle, okay? At the Golgi, at the lysosome, there is a transport that is facilitated by the vesicles. So this is to show you the dynamics of what's going on. The ribosomal proteins or even the histones are all imported into the nucleus, particularly the ribosomal proteins. They localize to the nucleolus, which is here in heavier blue, and they bind to a pre-ribosome RNA. And this process leads to a cleavage or maturation of a pre-ribosome RNA, and then a final procedure that will binding ribosomal RNA with ribosomal proteins that will generate the, I can localize the arrow. Okay, I can generate the 18S pre-ribosomal particle and this sedimentation ribosomal particles. S comes from sedimentation, meaning that it's not a measure of density, but it's a measure of where they sediment when we run an ultracentrifugation process. So we subject these particles to the ultracentrifugation, like a very, very fast spin, and then they will eventually localize to one position in that, or one band in that sedimentation. And that is what it means. 28S is that is more at the bottom than the 5S, okay? And there are other particles, ribosomal RNA, that come from outside of a nucleolus. Not everything comes from the nucleolus. So sort of this RNA comes from outside. And then they are exported through the nuclear pore. So this is a very, very interesting example about export. Not everything is import into the nucleus. There is also export. And the export, for example, the ribosome particles that finish the maturation process outside the nucleus in the cytoplasm, which means this will bind to, this is the 40S subunit, and this is the 60S subunit. These two will assemble together together with the tRNA, the transfer RNA, and the messenger RNA, and will be responsible for translating the protein. Okay, all the genetic information passes through these mature ribosomes. All right. So this is the nucleus. It contains chromatin in the form of euchromatin, typically 90% of the genes in this Lec configuration. Heterochromatin, very compact and binding to the periphery, to the lamin and to the inner envelope. The percentage may change. For example, I told you about the stem cells. They have more heterochromatin, more euchromatin, because it's more silent, it's less silent, and it produces lots of different proteins. Then we have the nucleolus, where the RNA processing occurs with mRNA splicing, rRNA maturation, and assembly to form the ribosomal proteins. There is import, there is export, nuclear envelope, two consecutive membranes with distinct composition and function. For example, the lamin will be found always binding to the inner nuclear envelope. And the ribosomes, for example, will be found always binding to the outer membrane, okay? So we have to make that distinction. They are not identical. All right, so we have, and this is exactly what it's been said here. The lamin or the lamina, the inner part, when mitosis comes in place, the lamin gets phosphorylated and disaggregated, and with that, the nuclear envelope gets disaggregated. So that's the molecular explanation of what's going on when mitosis ensues. Okay, the nuclear envelope has to disaggregate, and the driving force is phosphorylation of the lamin. Okay, and the outer membrane associated with the ribosomes. Nuclear pores, bidirectional gating. Okay, import and export. Very large protein complex formed by 50 nucleoporins. Okay, each of the nuclear pore is made out of 50. It's a complex of 50 proteins. Very complex, with a basket, asymmetrical, anchored to the envelope, etc. There are 3,000 to 4,000 pores in the nuclear envelope. The nuclear pore is very active. For example, 100 histones pass through the nuclear pores every minute. So imagine all the dynamism that is involved here. Okay, small proteins pass through diffusion. No need to ATP, no need to energy. But large proteins require active transport via chaperones. You remember the importins, GTP and ATP hydrolysis. When ATP is hydrolyzed, it releases the third phosphate, and there's a release of energy, and that is how this is being measured. Nuclear proteins bear nuclear localization signals. Okay, if there is no nuclear localization signal, the protein will not be imported. There are experiments, the original experiments that demonstrated the presence of a nuclear localization signal. How would you think that one experiment could be done to demonstrate that. Essentially, either you remove the signal and the protein will not be imported. You can mark that protein with GFP or any other marker, and then notice that that protein does not come in, the protein without the NLS. And the opposite experiment is to add a nuclear localization signal to a protein that is not fated to go to the nucleus, and you will find it in the nucleus. So all the information is contained in this sequence of, I don't know, 4 or 5 amino acids. I'm just making up the number, but very, very few amino acids. That's the driving force for importation, okay? And if you want to demonstrate that one of the importance, the chaperones is required for the import, what is the experiment you need to do? You need to mutate that imported and eliminate it from the system and check that the protein is not in. It doesn't get imported. It's a little bit of my flavor in research, okay? So every time you need to demonstrate requirement, you need to eliminate that sequence or that protein from the system and see what's going on. All right. Mitochondria. So first of all, it's been originated from a bacterium. You remember that, okay? And that's one of the main features of an eukaryotic cell. So what do you have here? You have, again, 2 membranes, the outer membrane here and the inner membrane. If you think about it, when a bacterium has been phagocytosed, you would have one membrane from the cell and the other membrane from the bacterium, right? So these two membranes are completely different. And the inner membrane, so the outside or the outer membrane comes from the cell, and the inner membrane comes from the bacterium, okay? All the processes related to making or producing energy will be located in the inner membrane, because that membrane comes from the bacterium and it contains all the complexes that are required for the production of energy. Okay, so there's a lot of logic in when you look at the mitochondrion, you will see 2 different membranes, and you can distinguish one from the other because the inner membrane looks more like a bacterium, and it has all the complexes that are required for the oxidative phosphorylation that will make the ATP, will make the energy of a cell. So this is a structure with invaginations denominated crista here. All the complexes that are the cytochromes, the dehydrogenases, and flavoproteins, okay? All these complexes, complex 1, complex 2, complex 3, complex 4, complex 5. These are very, very large complexes, and they're required for making ATP. It sort of utilizes electrons to each time to have lower energy, and the system, very smart system, transfer the energy from the electrons to create a new bond in the ADP to create ATP. And ATP is the energy because it's been used for every process. So this is the process of respiration in the mitochondrion, which is very similar to the process of respiration of us. We consume oxygen to create uh, CO2 and energy to respirate. The mitochondrion does the same thing. It gets the oxygen and a glucose or any or a fatty acid or some other molecule that has a lot of energy, and it creates CO2 plus ATP. So that's a typical process of respiration or combustion, the same thing. What happens here, it happens at the mitochondrial level. So we have also, so these are the, I'm sorry, I cannot really use well the pointer. Okay, we have the complexes here, the membrane of a crystal, the inner part. We have intracrystal space or matrix where the Krebs cycle is being completed. Okay, that's the first part of the obtention of the ATP. That occurs or takes place in the matrix. Then the second part takes place in the inner layer of the mitochondria, okay, where all the complexes are. All right, and there are mitochondrial granules, there are cation binding sites, etc. There are lots of different things, ribonucleoprotein particles, and it's also worth it to mention that the mitochondrion has DNA there. It's a very old DNA, and DNA, what the remnants of DNA from the bacterium. And they're responsible for making all these complexes of the mitochondrion that will generate the energy. Okay? And however, all these, the proteins, the mitochondrial produced proteins are not sufficient to provide all the, or the DNA of the mitochondrion is not sufficient to provide all the proteins that need to be in this structure, in this organelle. So many of the proteins are being imported. You remember, as an example of post-translational import, they come from outside to the inner part of the mitochondria. So some of the proteins are produced within the mitochondrion, very few proteins related to the Krebs cycle and oxidative phosphorylation, but many of the proteins are coming from outside. So they are coded at the nucleus in the genomic information of the nucleus. This is how they look, and you will find the mitochondrion very close to other structures, particularly because when you see a complex of mitochondria, and remember, structure leads to function, you will instantly realize that there are energetic processes or processes that require energy. So that's very important. For example, here, you see a lot of ER complex or structures. So apparently here, the ATP that is produced or created in the mitochondrion is going to be important for some unknown processes that will take place at the ER. So there are lots of we can predict lots of different functions based on the structure of the different organelles. Location. The form or the structure follows function. Okay, for example, these are examples of a kidney tubule cell with accumulation of mitochondria in the area that is required for ion exchange. It requires a lot of energy. So why having the mitochondrion elsewhere? It's better to have them in situ, in the place where the energy is going to be produced. So recapitulating, the mitochondrion contains its own DNA that produces enzymes involved in ATP formation. Another set of proteins is imported. Protein import occurs post-translationally. ATP and chaperones are required for protein import, okay? And one might say, if I require ATP for the protein import, why do I produce ATP? There's no net change there, but in fact, there is a lot of net change. The production of ATP is much higher than the usage of ATP, okay? Usually is 32 molecules of ATP for one cycle of the Krebs cycle and oxidative phosphorylation. Okay. Then there is another organelle that uses oxygen. So the mitochondrion uses oxygen, right? As I mentioned before. The peroxisome, it also uses oxygen, but that consumption is not coupled to ATP production like in the mitochondria. Okay, so it's a little bit different, and I'm gonna tell you a few things about the peroxisome. Why is it for, etc. Okay, importance of cytochrome C. First of all, this is the structure of a complex at the inner mitochondrial membrane, and how the electrons that have very high energy in complex 1, they start losing energy, and that creates a proton gradient within the inner membrane, from the outside of the inner membrane to the inside of the inner membrane. The proton or the hydrogen gradient then sort of gets into the matrix, and that produces the ATP, okay? So these are concepts that you've been through before. I just want to give you like a slight overview of those and put them all together. Okay, and then we have oxygen consumption and ATP production. There's one molecule, it's a small molecule, cytochrome C. It's very interesting, and that happens in research. You identify one protein with function A, and you think that that's the only function. But in fact, the system is very versatile. One function may lead to another function, and the protein may have 10 different functions. For example, P53, which is a proto-oncogene, it's called the guardian of the cell, and it's involved in 1,000 different functions, okay? So it's not that it's one-to-one. There is a lot of different functions ascribed to one protein. So this is one case, cytochrome C. Cytochrome C is a molecule, a very, very small peptide that goes back and forth from complex 3 and 4, and it's loosely attached to the membrane, right? So what happens when the cell is damaged? Let's say it goes up, it is injured by any reason. For example, in the muscle by contraction, etc. So that's an intrinsic injury. The cell has to go, has to apoptose. Apoptosis is programmed cell death, right? There are extrinsic signals coming from elsewhere. They tell that cell to disappear, to apoptose, or there are intrinsic signals like the cytochrome C. Now we're going back to the cytochrome C. Cytochrome C, which is loosely attached, if there is some damage in the mitochondrial membrane, the cytochrome will migrate to the cytoplasm and will complex with the pre-established complex of apoptosis, which is called the apoptotic complex, right? And this will trigger, it will activate the apoptosome or apoptotic complex, and that complex will eliminate that cell. So that's a new function of a cytochrome C. Okay, it's not just transfer of energy. But keep that in mind, all these different functions that they interact one to another. All right, we're going to move into the secretory pathway. As I told you before, this is a characteristic of an eukaryotic, a mammalian cell, for example, right? And if you see, for example, these vacuoles loaded with certain complex of proteins or glycoproteins, that to us, that it's clearly a secretory cell that will accumulate as much zymogen or other complexes, waiting for a trigger, waiting for a signal to release all those complexes to the exterior, extracellular phase of that tissue. Okay, so and the way it works is the system, the eukaryotic system, organized itself to create a mono or a bidirectional mechanism of secretion. Okay, in this case, it's being provided by the ER, the endoplasmic reticulum, which in other tissues, it's called slightly different, like sarcoplasmic reticulum, but it's essentially the same thing, okay? The endoplasmic reticulum, and then the ergic, the intermediate ER Golgi, and finally, sorry. And the ergic, and finally the stack of pancakes that is very typical when you look at the microscope. That's the Golgi, okay? And the Golgi is responsible for sort the different complexes, the different proteins. Essentially, it will say, you go that way to the lysosome, you will go to the membrane, you will go back to the ER, okay? Is the main traffic sort of organizer. That's the Golgi, okay? And to do that, it needs to add into the basic proteins that are being formed, it needs to add some glycoproteins, for example, some glycosides to make a glycoprotein that will protect that protein from being degraded once those complexes are being secreted outside. Outside of a cell, you are in a very dangerous world because there are lots of different proteases and glycoproteases, but the glycosides are protective. They really protect the protein that has been secreted. So if you see, for example, a protein from outside of a cell in the extracellular matrix, it will 100% or 99%, it will be made also by glycosides, okay? It's gonna be a glycoprotein. All right, so, and here we have, so most of the cell sorting occurs here at the Golgi. And they can go to the plasma membrane, they can go to the lysosomes for degradation, so it may play different functions. Okay. Let's look at the endoplasmic reticulum, the first component of a secretory pathway, the ER, okay? So this is the nucleus here, and this is the nuclear envelope. And as an extension of a nuclear envelope, you will have the ER. So the ER is not completely independent from the nucleus. In fact, many of the proteins that are located at the nuclear envelope will migrate from inside of the ER, and then they will pass to the Golgi, they will advance to the Golgi, okay? So the lumen is quite shared between the nuclear envelope and the ER, okay? And you can see here the ribosomes that are attached only to the outer nuclear envelope. Here you see the ER, the RER, the rough endoplasmic reticulum, with ribosomes on both parts of the membranes. Okay, there's no outer and inner because they go in a disorganized way. They're not like the nucleus where you can identify one outer and one inner, but you could see ribosomes in both membranes, and that's very distinctive. If you have, for example, one sample that is nuclear and one sample that is cytoplasmic, and you see ribosomes attached to a membrane, you will be able to identify, to distinguish them. In one case, the ribosomes will be attached only to half of the membrane. In the other case, you will see ribosomes attached to the whole membrane. Okay, that's very important. But always the outer portion. Okay, for example, the lumen, you will not see ribosomes inside, the same way that you will not see ribosomes in the lumen of a nuclear envelope. Ribosomes. The ribosomes, as shown before, they start at the nucleolus. Okay, the ribosomal proteins, they are RNA, they assemble into pre-complex, then gets mature, then they get exported, and they assemble once they see a messenger RNA that will be the template for a protein production, and the transfer RNA that will contain each of the amino acids that will be added to the nascent protein one after another. So all these ribosomes, one may think, yeah, but one ribosome will be attached to the RER, and one ribosome will not be attached, will be in the cytoplasm, right? And that's true, but they both come from the same pool. So you have a pool of ribosomes here in the center, and those will form the cytoplasmic proteins, and also that same pool will form the proteins that are attached to the ER and will be eventually secreted to the extracellular matrix. And one might think, how come they're not different, these ribos? They look for differences for years, the scientists, and they've never seen any different. All they saw is that the ribosomes were attached to the ER or not, but no molecular difference. What's the difference? The difference is being provided by the sequence of the nascent protein. The nascent protein has a signal that will be recognized by a complex and will produce a channel within the ER and allow the protein to be transported into the lumen of the ER. Okay, so that's the difference. It's not the ribosomes, it's the protein, right? And since that occurs, all this process of importing to the ER occurs at the same time, this is called a co-translational import into the ER. This is the typical co-translation. I'm not sure if there's another one. This is probably the only co-translation. There may be, but there are very few. Most of the imports are gated imports in the nucleus or post-translational import, but not co-translation. But this is a typical example of a co-translational import. And usually they act the ribosomes. One ribosome catches one messenger RNA. And then automatically there are more and more that join that messenger RNA to make a protein. And they form what it's called a polyribosome. It's not, you will never see a messenger RNA with just one ribosome that is being sort of scanning and reading the sequence. You will see like a mix of many ribosomes there. And at the same time, simultaneously, you will see the nascent protein being produced. In this case, they don't have the signal for the ER, so they remain in the cytoplasm. But these ones contain a signal here in red. You can see the red here, ER signal peptide. So they attach to the ER, they form this cavity, and they are being co-translational imported into the ER, okay? And this is a schematic showing the whole complex assembled with the two subunits of a ribosome, the 40X and the 68, and the 60S, sorry, 40S and 60S with this is a very transfer view where you can appreciate the tRNA, the transfer RNA, the one that brings in the amino acids, the messenger RNA, the template, which brings the coding sequence, and the protein, starting with the amino terminus and then completed towards the carboxy terminus. And the cavity has been formed because of a signal that is contained within the protein sequence, okay? This is just to show you how they look at the free and bound ribosomes. All these are free in the cytoplasm. They hang in the cytoplasm, and all these are bound. And many years passed when they try to see a difference between ribosomes, never been able to see anything. And finally, the sequence, the driving force was in the sequence of the protein. So this is for endoplasmic reticulum. There is a soluble and ER ribosomes. They are identical. An ER signal sequence in the protein is recognized by a signal recognition particle, okay? SRP, which allows for a co-translational translocation of a protein into the ER. So at the same time, you'll see the protein being created. At that same time, it's already going into the ER. It's co-translation. Hydrophobic proteins are anchored to the ER membrane. They have the signal recognition, and also it's more hydrophobic. Hydrophobic means that they hate the water or the cytoplasm, and they love the lipidic structures, right? It's hydrophobic versus hydrophilic, okay? So if the protein that is being produced or synthesized is very hydrophobic, it will try to go in, but it will be retained by the ER membrane because it's made out of a lipidic kind of structure, right? A lipidic composition. It'll be retained. And those proteins may probably be those ones that will go and will be anchored to the plasma membrane at the periphery of the cell. The ones that are not hydrophobic, they are hydrophilic, they will go all the process with the ER, the Golgi, and then the vesicle to be released into the outside space, okay, the extracellular space. All right? All right. A precursor oligosaccharide is held in the lumen of a ER membrane by a lipid named dolicol. So as I told you, at some point, the protein, whether it's been anchored or secreted, it needs more glycosides. It needs more carbohydrates, okay, like glucose, mannose, and a chain of glycosides that will protect the proteins from being degraded. And it's been provided at the first stage from the dolicol that will transfer, for example, glucose from one place to the protein, or mannose from one place, from the membrane to the protein. And so protein, where are we? Protein glycosylation takes place in the rough ER almost at the time the protein is being translocated, okay? So it's been translocated and glycosylated at the same time. Proteins fold and oligomerize, but misfold, there has to be a checkpoint at some point, okay? And misfolded proteins are somehow translocated back into the cytosol, to the cytoplasm, and they are being degraded by the proteasome. The proteasome will degrade all misfolded proteins, okay? So they have a sensor. They say this protein cannot advance, has to be translocated back into the cytosol, and then we will degrade it. Incompletely folded proteins do not advance to the Golgi, okay? There are checkpoints all over the system. Examples of specialized functions of the ER, the one that stores calcium is called ER, the muscle ER, the muscle ER, which is called SR, the sarcoplasmic reticulum. It stores a lot of calcium and it releases calcium at the same time that will facilitate the contraction of the skeletal muscle, okay? So it's very specialized. There are many others. Another example is the one that is involved in detoxification of hydrophobic toxins, like the patocyte, ER, okay? Okay, we'll move into the Golgi. So this is where it is located and how can it be identified? It's a stack of pancakes. It's one pancake after another. This is the Golgi. You will not see a structure like the Golgi, okay? And it's very dense, has a lot of surface, but very little volume, okay? Same with the ER. The ER is all over the cell, and it's very dense, a lot of surface and very little volume. Okay, this is how it looks. And these are the different components. The Golgi proper is this one in blue, but before there is an intermediate ER Golgi, okay? They're composed of both structures. It's the one that connects the ER with the Golgi. And it's called ERGIC, ER Golgi intermediate something. I don't remember what it is, okay? But it's intermediate. That is what matters. Then we have the Golgi proper with the cis phase, the medial phase, and trans phase. Why is this important? Because the Golgi plays an important role in cell sorting, and it does it consecutively. So it starts at the cis Golgi, then if everything is done, then it advances into the medial Golgi, and finally into the trans Golgi. So, or the trans phase and then the trans Golgi. So it is very, very well synchronized. And in each portion, there are distinguishable activities. And it ends up with a mature glycoprotein. And that glycoprotein will go either to the lysosome or to the plasma membrane or to the extracellular matrix. And how this stack of pancakes is connected through what you can see here, vesicles, okay? Vesicles that are spherical or they could be tubular. A bunch of spheres all fused together and they go from the ergic to the Golgi, okay, as one tube of structure. So they could end up with smooth or coated vesicles. Coated means that it's coated by proteins. Smooth without coating, sorry. And they could be secretory granules as well. All these things that are going to be secreted, they are born in the Golgi. The Golgi, the ER forms the immature glycoproteins, adding glycosides or carbohydrates, but then that structure is being converted or transformed. Some carbohydrates, for example, originally it's made out of mannose or glucose. Some mannose are eliminated and replaced with more complex glycosides, okay, or chain of carbohydrates. And those complexes are being exemplified by acetylglucosamine, which is a carbohydrate that's been added. So it grows in maturation, in complexity. Galactose or acetylnuaminic acid or NANA, okay? So these mature complexes are being added in the Golgi. So the process starts in the ER and the final maturation takes place in the Golgi. So the Golgi forms glycoproteins as well as the ER. You will have more details of these processes, but these are the concepts I want you to retain. And this is the sequence of events. For example, one protein has very immature carbohydrates that are phosphorylated, and then as this glycoprotein advances, there's going to be a removal of one carbohydrate, mannose, and it will be replaced by NANA, which is much more complex, okay? Or even if it's not replaced in another sequence, in another amino acid, it'll be bound another complex, another carbohydrate. Then addition of galactose, and finally in the trans-Golgi, addition of NANA, okay? So it's a very distinguishable process. And then there'll be lysosomes or secreted vesicles that come out of the Golgi. And the protein itself now contains information where it needs to go, but also the vesicles, the coding of the vesicle is also telling us where this complex will go, where it will end up. And I'll give you a couple of examples about that. In some cases, for example, let's say that the protein went through all the checkpoints and so there's a liberation of a vesicle that buds off the Golgi, and it contains newly synthesized plasma membrane lipids. For example, it's not just about proteins. It could be lipids that will go to a membrane and will restore the membrane content, etc. And in some cases, this system is constitutive or undergoes a constitutive secretion. There's no signal, there's no trigger to be to fuse, to dock, and to merge with the membrane. Some cases it's constitutive. In some cases, the proteins are waiting online here, waiting for the signal to happen and to trigger the process of the merger, okay? To the docking, recognition, and then the opening and secretion. For example, signals such as hormones or neurotransmitter. For example, the mast cell contains a lot of histamine, but we don't need the histamine all the time, only when there is an allergic reaction, right? So all these mast contain those vesicles. They do not trigger the release of the histamine only, or except only when there is a signal, like, for example, histamine. Histamine will bind to a receptor and it will trigger the signal. Okay, now you can release your product. So that is not constitutive. It's regulated secretion. Okay, so you have all the options, all the different flavors. Okay, Golgi. Vesicles destined to go to the ER, to the Golgi, are COP2 coded. So the code, the protein code of a vesicule also is telling us information, it's giving us information. When you see, for example, a code that is COP2, that means that goes from the ER to the Golgi. And when you see vesicles that are COP1 coded, that means that it undergoes a reverse transportation from the Golgi to the ER to replenish the membrane that has been used, okay? So it has to go both ways. Secretion and then replenishment. Okay, vesicles destined to go from a Golgi to the plasma membrane are clathrin coded. Clathrin is very typical code that will inform the vesicle to go to the plasma membrane, okay? In the Golgi, formation, processing, and maturation of glycoproteins take place. Addition of oligosaccharides to the protein makes it more resistant to proteases when they are secreted, okay? In the ER also, the process of addition of simple saccharides is also taking place. Vesicles are transported by motor proteins, and we'll see that example. Dynein or kinesin, it's not random. They just don't go into the void to the and crash to the plasma membrane. The process is very, very well regulated. They walk through motor proteins, through the microtubules, and they dock at the plasma membrane. This is a good example. Phosphorylation of a mannose, like mannose 6-phosphate, phosphorylation of this residue targets a protein to the lysosome. For example, a hydrolase. Lysosome has a lot of hydrolases that will proteolyze lots of different proteins and many other components. But essentially, this process, the mannose 6-phosphate, will protect the hydrolase from being degraded beforehand or from degrading proteins beforehand. They need to get to the lysosome or to the late endosome to be active. If they're active before that, that would be a problem for the cell. So the phosphate at the mannose is sort of stopping or putting a brake at the hydrolase, and it's telling the hydrolase, stop, don't start now. Start when the phosphate is liberated. And here's what happens. Phosphate in the late endosome or in the lysosome, the pH, it's very acidic, and when it's very acidic, the phosphate is released from the mannose. And once the protector is being released, then the hydrolase will act. And that's how this happens. And there is an example that I'll show you in a minute. Okay. Orderly process in vesicular trafficking, budding off, docking, fusion, okay, the merging or the fusion. Several proteins are involved in this highly regulated process. It's never random. The vesicle, when it leaves the Golgi, it knows where it should go already. It has all the information, okay? Secretion to the exterior can be constitutive or regulated, like histamine, the example that I gave you before. So this is the opposite to secretion. This is endocytosis, okay? Has to be both ways, always. Endocytosis or cellular eating or cellular drinking can occur like fluid phase endocytosis. So essentially, the cell will endocytose whatever is next to it, the liquid, the extracellular matrix liquid, with the components in the liquid, okay? So that's a typical random endocytosis. But there are other processes that require receptor, that they are receptor-mediated endocytosis. For example, the cholesterol. Cholesterol is endocytosed into the cell with the LDL receptor, using the LDL receptor. Okay, this process is programmed. It is not constitutive. And then we have phagocytosis. It's a solid. It's when the cell, a professional, for example, macrophage, will see a bacterium, huge cell there, bacterium, but it's smaller than the eukaryotic cell. So it needs to eat it. That's the function of a macrophage or a neutrophil, okay? These are professional eaters. And they eat, for example, bacterium or other particles, okay? And if you can compare these, for example, the size of these particles could be 0.1 microns, while for a phagocytosis of bacterium, they could be larger than 1 micron, okay? So there is a difference, a big difference in size of these processes. But essentially you have endocytosis and phagocytosis. So eating and drinking, endocytosis, constitutive or receptor mediated, like LDL. Phagocytosis performed by professional phagocytes, like macrophages, dendritic cells. These are all immune cells that they say, okay, there's an alien here, we have to eat it before it starts reproducing and making more of those, all right? And this process is very quick. It has to happen within seconds. Then there's another process, which is autophagy. It's self-eating. Sometimes, for example, mitochondrion is being damaged, okay? And the system will evaluate what is best. Shall we kill the cell? Shall we apoptose the entire cell? Or we eat specifically the mitochondrion? or we eat another organelle, for example, let's say part of a Golgi, or ribosomes that are not working well, okay? And this has to happen very quickly. And autophagy can happen with this organelle. Sometimes it's more efficient to eat just one of the components, not the entire cell. Okay, so the cell manages to eat only one component. And the process, particularly the process of eating the mitochondria, has been coined, and it's called mitophagy. It's like autophagy, but autophagy of the mitochondrion has been coined by mitophagy. Okay, it's a very typical process. It's an orderly process with early, late endosomes and lysosomes. Okay, that occurs in a consecutive way, and the pH, the concentration of protons, the pH, becomes more acidic as they go. The lysosome is the more acidic one. It's pH 5 or so. Okay, the cell has, the cytoplasm has a neutral pH, like 7.4. So it's 2 orders of magnitude in a cell is critical. For example, if a phagosome, if a lysosome is broken or is damaged, and the pH 5 may kill the cell if it's not treated properly. Okay, so at pH 5, the phosphate that is bound to mannose is being removed, and the hydrolases are working very actively. So, and the pH is being decreased, goes to more acidic as the system passes from early, late endosomes and finally the lysosomes. Some endosomes undergo transcytosis, meaning that they don't degrade. They just, for example, they endocytose a few products at one side of a cell, and they go all the way down to the other side of the cell, and then it gets released. So what it does, it transports some products all across the cell, right? That's transcytosis. There's no degradation involved there, or minimal degradation. And some, they just degrade to secrete, or it's called also defecation, okay, where it secretes products that are not going to be used anymore, right? The lysosome gets rid of many of the products. Some endosomes, oh no, this I already mentioned. Okay, there we go. And this is an example of bacterium being phagocytosed, end up in the lysosome, or endocytosis here through the different phases, early, late endosome, lysosome, and then autophagy or mitophagy if the load is the mitochondria. This is an electron micrograph. There's in cell biology, everything is about electron micrographs. They'll instruct us on what is this about, okay? This is an endosome. This is the clathrin coat that I told you. They have less energy when the clathrin, so there are all these different isomers, all these clathrin subunits. They cover the inner part of the membrane, right? And they have more energy, or they stabilize at an energetic point when the clathrin sort of curves in this way. right? So it's all about energy. If you give them more energy, it will normalize the bend. But if you remove energy, they will sort of mature into a vesicule, into an endos, like this, okay? So the Clathrin code is the driving force for the creation of the endocytosis or the vesicule here. And there is one interesting experiment that I usually give it in the in this class, which is how regulated is the endocytosis of cholesterol, for example, LDL, or how synchronized to the Clathrin protein is this process. And usually, and what it is, there is one scientist who did some experiments with the receptor. Essentially, when the receptor is intact, the LDL, the cargo, will bind to the receptor, and then you will have the Clathrin that will bend because of this recognition of sites and will proceed or advance into the vesicule, okay? But in this case, you will have the Clathrin, but it will endocytose without any load, because these receptors have been genetically manipulated, and the cytoplasmic part of a receptor had been taken away. And since there is no recognition between receptor, load, and Clathrin, this vesicule will be just unloaded, okay? So the genetics can explain lots of different behaviors. Okay, the lysosome. The lysosome operates at pH 5, and the cytosol is at 7.2. So there better be a good separation between these two pH compartments. Essentially, to reach a pH of 5, there has to be a lot of energy that comes again through the ATP and proton pump in to the lysosome. So this excess of proton is the one that gives the right pH, okay? And there are acid hydrolases, nucleases, proteases, glycosidases, lipases, phosphatases, sulfatases, phospholipases. Okay, these are examples of all these proteins that can be used. This is an example of how this operates. So essentially, you have a protein that has a mannose and a phosphomannose at the ER. And then the phosphate, so this is covered by Clathrin, and it's been recognized by the M6, the mannose 6 phosphate. And it goes all the way through here, and the pH acidic will allow the phosphate to be released, and this receptor will be returned to the Golgi and to the ER. Removal of the phosphate will indicate that this hydrolase can be active or can work. And this happens over and over again. Peroxisomes that I mentioned a couple of things at the beginning. is responsible for the catabolism of fatty acids, removal of toxic compounds, hydrogen peroxide production. Once it produces hydrogen peroxide that has a very short life, it will kill microorganisms. It'll be responsible for detoxification of alcohol, etc., etc. And they look very refringent to the electron micrograph. And as opposed to the mitochondrion uses oxygen to generate ATP. The peroxisomes uses oxygen, but does not generate ATP, okay? So that's a clear difference between these two organelles. Here's how it looks. Peroxisome is very dark, very refringent. You could see mitochondria here, you could see lysosomes here, etc., etc. Okay, and now we look into the cytoskeleton. The cytoskeleton it goes into the organelles lecture, not because it is an organelle, but it's because the organelles can be transported via the cytoskeleton. For example, the microtubules or other filaments. Okay? And that's why we include the cytoskeleton into this overview lecture. And we will have more details about the cytoskeleton. So essentially, there are three types: the actin, tubulin, or vimentin. Actin for actin filaments, tubulin for microtubules, vimentin, lamin, for example, the one at the nuclear envelope, that is an intermediate filament or viment. Fibrous proteins for intermediate filament, okay? For microtubules, you have, for example, the mitotic spindle. That is a clear example of microtubules. And the motor proteins that will take the cargo proteins to the plasma membrane, for example, they sort of walk or run through the microtubules. And the actin is more responsible for the periphery of a cell and is responsible for maintaining the shape of a cell. So actin filaments maintain the shape of the cells and its polarity, control cell migration, etc. And it's typically localized to the periphery of the cell, the inside of the cell, but the periphery. And it's the one responsible for example, if a cell needs to migrate as a whole, the actin filaments are the ones that are driving the cell towards that direction. So these are the actin filaments, microtubules, star-like structure attached on a perinuclear centrosome. So there's a centrosome, and then structures, star-like, that are going like a star. And they are like the template of a scaffold proteins, like dynein or kinesin, to position the organelles in the cell, okay? Like in the ER, Golgi, etc. Intermediate filaments, they will provide from strength to the cell. These are like these intermediate filaments that are the ropes of the cells. They maintain the mechanical stability of the cell. And this is how they look like. These are the actin filaments here, located at the periphery, and they are responsible, for example, they attach to the cilia, which are responsible for the movement of the cell. So this is how they look, the actin filaments. Then we have intermediate filaments, the ropes, for example, vimentin or other intermediate filaments that will bind desmosomes or desmin, etc. All those will bind cell to cell and will make the complex of the cells much more stable. And then the microtubules. The microtubules are hollow they have void inside, but outside they are very strong. And you can see that the microtubules are formed by alpha and beta tubuli. They sort of organize themselves in a very geometrical structure. And we'll see a couple of things. So this is for actin. Actins are very dynamic. They form filaments, but they also exist as monomers. And they can nucleate and be part of the elongation complex. All right, the G-actin for glomerular actin will nucleate and will form the filamentous actin. Okay, and this is like the treadmill situation in which there are monomers that are being introduced and there are monomers that are being released. And this, you can start the treadmilling, walking through those filaments, and you will see the disappearance on one end and the appearance on another end. And you reach a steady state. And that happens with the actin. Of course, there are molecules or proteins that modify the rate of incorporation, like, for example, thymosin or profilin or gelsolin. They will modify the incorporation of the monomers into the filament. Okay? Here, for example, you have the addition of profilin will add or will make this process faster. So the treadmill will start growing from the very beginning, okay? Then we have, for example, molecules that retard the incorporation of the monomer into the filament, like, for example, thymosin complex. And there are other proteins that are responsible for breaking, for severing the actin filament, and that is called that is called severing and capping. Okay, so it disassociates two actin molecules. This is for example, desmin or lamin. There are intermediate filaments. They bunch one to another all together. They bind to the DNA, they bind to other proteins, they bind to the inner portion of the nuclear envelope, but, and this is very strong, but when there is mitosis, there is dephosphorylation of lamin that disaggregates lamin. The whole nuclear envelope is disaggregated and therefore will proceed with mitosis, okay? So just a phosphorylation is a trigger of the entire disaggregation of a nucleus. This is another example of intermediate filaments with, for example, desmoglin, desmocolin, placoglobin, desmoplakins. There are many intermediate filaments that will tie up two different cells and will make it more resistant to mechanical damage, for example. This is for the beta and alpha tubulin that will be geometrically incorporated into the tubular structure here, alpha and beta, alpha and beta, alpha and beta, okay? And will form like a hollow molecule, but it's a huge molecule. And there, all the runners, all the walkers, the motor proteins will be walking through, okay? For example, this kinesin will be sort of walking through the microtubule stationary. It could be kinesin going from the minus to the plus end. It could be dynein, they could go to the plus end to the minus end. They have very specific instruction where they go. and they are loaded with the cargo, with the proteins or with the lipids or with the sugars, okay? So that is quite intriguing how these organelles are being placed through the microtubules. The typical example, the mitotic spindle that you see here, and it's very dynamic. The microtubules disaggregate, aggregate again, and when time comes, disaggregate again, etc., etc. Okay, so I believe this is the end. Yes, and next Wednesday, Dr. Ivesa will lecture. Will be in charge of a lecture. He will also explain a little bit about the exam, how it's going to be, and just follow as much as you can my suggestions. Try to study as soon as possible, and I'll be happy if you can email me questions today or tomorrow. Okay? All right, thank you. Just give me 1 second. Let me finish the recording.